Good afternoon. I would like to first thank the organizers and Kevin, especially for the invitation to participate in the symposium. It is a pleasure to be back at Iowa State, even if it is virtually. So thank you very much for the invitation, and thank you to all the organizers. So today I'm going to talk about breeding for the future and how do we put it all together, right? So how do we get there for breeding for the next maybe 100 years? But to start that, I'm going to start with what happened about 100 years ago. And what more suiting that starting a presentation at Iowa State with a picture of Henry A. Wallace. So about 100 years ago, the first hybrid maize was sold in the US, and that was from a company that Henry A. Wallace funded. And so, so you can see in this picture, most of you are probably very familiar on how the yield increases started after that momentum where we started breeding for hybrids and we started selling hybrids. And it's not all breeding, it's also management, but breeding had a huge contribution. And at that time, about 100 years ago, the main breeding target was yield. And this was grain yield, it was biomass yield, depending on the crops. And the main goal was food, or eventually food. We also had feed and fuel, but it was mainly to feed the population, the human population. But we now face more challenges. We face a growing population. The world population is increasing, and we're going to need to feed it. People are demanding more nutritious and high quality food, so we'll need to provide that. Also, we face this at the same time that there are more extreme weather patterns and we have climate change. So we are seeing more variability in the weather patterns. We're seeing more erosion due to the agricultural practices that we have used. And we need to guarantee the profitability and the sustainability and welfare of the farmers too. So all of these challenges will need to be met right now and in the future to accomplish our breeding goals. And so how do we get there? So I'm gonna talk a little bit about breeding efficiency, speed, and precision, and how can we get there? And so some of the examples from our research, and then how to tackle the multi-trait or multi-dimensions of this problem too. So first, I'm starting with the breeder's equation. So if we have a random mating population, like that one in blue, it has a mean, and now we select individuals that are, maybe truncating selection. We select individuals that are higher yielding. The difference between the mean of the selected individuals and the mean of the original population is what we call the selection differential. But how much of that selection differential do we realize in the next generation if we randomate the selected individuals depends on the heritability or on how well are we evaluating the genotype of those individuals and how much of that variability is due to the genotype compared to the environment. So in the next generation, if we randomate the selected individuals, the right tail, then we're going to see a progress, right? The difference between the mean of the original population and the mean of the cycle one generation is what we call the responsive selection. This equation can also be written as a function of the intensity of selection, that's the I here, the accuracy or how well are we evaluating those genotypes. So this is the correlation between the predicted and the true genotypic values, the square root of the genetic variances, and then also we can put this equation as a function of the time, so the time that it takes to accomplish one generation. So how can we tweak these to have a higher responsive to selection? And I'm going to frame this talk mainly in a breeding program that is either for producing inbred lines, so pure lines as in a self-feeding crop, or for producing hybrids where you need to still develop those inbred lines first. And so this is just a diagram showing first, we usually start with the selection of the parents, then we intermate the best parents, And then we need to somehow inbreed those, and we can use some haploids, or we can use selfing through several generations until we reach that. And then this cycle, which is our population improvement, continues, right? We select the next best parents and we continue improving our population. At the same time, we need to do the cultivar development, right? So we need to have yield trials evaluated in multiple years so that we can evaluate the individuals or the hybrids. We need to select the best ones until we can release the cultivars. If we're using some type of genomic prediction or other things, we can use this information with traditional also. We can use this information to select the best parents. So this is the cycle that where I'm framing my talk today on a breeding program. And so how can we increase speed, right? One of the techniques is double haploids, and this has been highly utilized in corn for a long time, and it's very successful, especially when we couple it with genomic predictions. Because if we do that double haploids and then we end up with thousands or hundreds of thousands of individuals that we need to phenotype, then that becomes very expensive. But if we can narrow those down with genomic predictions, then we can increase the speed at the same time as not overburdening our program. So this is helping. I'm putting here my breeders equation in the top, and it's helping with the time. We can also use this technique called speed breeding that is very fashionable these days. That is just old things such as controlling the temperature and controlling the light that we have known for several decades that work. In increasing the speed until flowering time. So we can force the plants to flower earlier by manipulating this photoperiod and temperature. So we did this in oats, for example, and we went from 111 days until harvest to 79. So that means that if we're doing this, trying to have it in a greenhouse, for example, small grains work very well in the greenhouses. We're trying to push faster cycles. We can do a single seed descent in the greenhouse, and we can have now 4 or 5 generations in a year. So this means that it's very fast for this process of inbreeding until we can select the next parent. There are a few things that in this process we need to still figure out is how fast can we cycle or recycle the parents? Can we select at F2, at F3, at F4, so that in our genomic prediction systems, we can incorporate them faster. So the sooner we cycle them, the faster the system is gonna be if we can maintain the accuracy. So that's one of the things that we still need to figure out. The other things related with efficiency and the precision is this selection of the parents, right? How do we select the best parents, or can we, instead of selecting the best parents, select the best crosses or parent, not just the parent, but the cross. So which one of this pair combination is the best one? And this is based on not just looking at the performance per se of an individual, but mainly looking at how is the progeny going to look like. And it will change the variance depending on what parents do you cross. And by teaching the variance, we can also change the top 10 of those progeny could be very different, even though the mean might be the same because the mean is just the average of the 2 parents. So this is one strategy we have shown in wheat, how this is highly efficient to actually use it in our selection. So that's one way we can be more efficient at selecting parents. The other one is how can we optimize our training population? So how can we have, again, a higher productive ability by choosing which individuals to include in the training population? We know there are tons of studies that show that the more related individuals are, the higher the productive ability. But then if we want to go with just the more related, we are decreasing the population size, right? So there is a balance here. There is a trade-off between decreasing the population size and having relatedness. So how can we find an optimum? And there were several studies that tried to look at this. with different type of optimization. What we did is we compared some of those strategies to a new strategy that we proposed, which is a stratified sample based on the relationship to the training, to the testing individuals, so the testing to the training, and also a priori identification of this structure or groups in the population. So by weighting and doing a stratified sample and then weighting the relationship matrix by the marker effects, what we were able to achieve is a higher predictive ability around here, this purple line. So this is one way we can optimize the training population. So this means that this was better than using all the individuals, right? So, so not always is it best to use all the individuals that you have available for building and training population, but you can optimize this. Other strategies have to do with how do we deal with all the data that we have, right? And one is looking at individuals, the other one is looking at environments. We know that the performance in all the possible environments or the infinite sample of environments might not be so meaningful for our breeding program because they might not be part of our targeted population of environments. So how do we optimize this training population for specific groups of environments that are our targeted population? We can use this G/E to help in that, and we can model the G/E to do that. So by modeling this G/E, by building mega environments and predicting within mega environments or predicting for the known G/E structure, we can improve the predictive ability. That was shown in a few studies. Also, what we can do is we can create this regionalization of our trials and of our trials to decide on how to best predict the performance in farmers' field. So how can we actually use all this information about G/E, maybe adding environmental covariates, so that we can target what is the best cultivar for a farmer in a specific location. So we are working on that with a small grains community. This is just a preview of what's coming, but we've been able to use all the historical information to create this type of information. So again, this is trying to be better at predicting the performance so that we can have a higher response to selection. Then the other thing I told you about environmental covariates, we can also use all the information available, right? We don't need to just rely on the phenotype, which is a great thing to do to predict the performance if we're interested in grain yield, use grain yield information. But we can enrich that data set with environmental covariates. And there are different ways we can do this. This is just one study that shows that by incorporating this environmental covariates using PLS, we were able to increase the predictive ability, so by a lot. So this means that sometimes year-to-year variability that are hard to predict, if we can have environmental covariates, if we can look at the historical environmental covariates year by year, we can improve the predictive ability, and we can also predict for common environments within a location. So these are some strategies that we can use to improve the response to selection, mainly by working on the predictive ability. I show you first speed, so working with a T with the time. Now I show you a few that work with the predictive ability. The other component is we can also look at the evaluation process. How do we evaluate our when we need to evaluate them in yield trials in multiple locations in multiple years? How do we do that? And the most common strategy by public land readers is to use common trials or uniformity trials that a set of small number of genotypes are tested everywhere in all the possible locations that we can have in the, with our collaborators. So we use highly replicated within a trial and also the same set balanced across all the environments. And that produces a high predictive ability of those genotypic means. I mean, we get very, very good estimates of those genotypic mean values, right, with that strategy. But is it the most optimal in terms of the response to selection? Remember, the response is not just a function of how well can we predict the performance of an individual, but also the intensity of the selection and the variances. So what about trying to balance a little bit more unbalance with having larger population sizes? So can we increase the population size at the expense of balance, maintaining the same resources to increase the response to selection? And what we've shown in this study is by looking at the experimental designs and looking at what is more optimal, we found that in terms of the predictive ability, the alpha, so these are resolvable incomplete blocks, are optimal. So these are replicated in each one of the trials and balanced across all the trials. So this seems to be working very good because we get replications, we get very good local control, and they do very well in terms of predicting the true genotypic values. But then when we look at the response to selection, we're limiting the number of genotypes that we evaluate. And we found that having some of the PREP or augmented PREP designs where we keep the same number of plots, the same number of experimental plots, meaning that we have larger population sizes, we were able to increase the response to selection. So even at the cost of replications. So this is something that is very important when we're thinking about evaluating this. How do we put all those resources into the most efficient way in the experimental designs? We then did the same or evaluated this on assuming that this was just for one location, right? But now assuming that we have multi-environment trials, right? We usually evaluate them in multiple locations. Can we unbalance across locations too? And how much can we unbalance so that it becomes more efficient? And so what we did in this other study was to compare, this is like the classical uniformity trial, where we have a set of 20 genotypes evaluated in every single location. So these are 6 locations as an example. Every single genotype is evaluated in every single location and with 3-fold replication. We compare that to what we call a balanced PREP. In this case, we have an augmented or a PREP design where we, in each one of the locations, we have some individuals that are replicated and some individuals that are unreplicated. So just once in the trial. But then we balance it across all the locations. So we did the same set of genotypes evaluated in all 6 locations. And then we compared that to a new experimental design that we proposed that we call the MED or Mega Environmental Design. And in this case, what we said is, what if we think about this whole set of trials as our experiment, right? Can we think about this as our unit? And therefore, if we have mega environments, within one mega environment, we'll treat it as a unit, and we will not replicate, will not put all the genotypes in all the locations. We'll have imbalance across locations, but because we assume that within a mega environment, the G/E is quite low, that's the definition of our mega environment, then we can assume that we can borrow information from the other individuals. We're also assuming that we have a known a kinship relationship matrix or pedigree information where we can borrow information from the different related individuals, right? And then by doing that purposefully unbalanced designs, we decided to evaluate 2 sets of replications within, because there's a lot of field variability within trial variability. This is micro environmental variability. We also put some replication within. And what we found when we evaluated this fully replicated and fully balanced across locations, then unreplicated or partially replicated, but balanced across locations, and then this mega environmental designs, we found that the response to selection could be increased by 40% when we purposefully unbalance across this. And this was done with real phenotypic information, real field variability, real G/E. So we were trying to reduce the assumptions, but it was all simulated data based on all those real data. So these are some strategies that we can use. In this case, what we're doing is we're playing with the intensity of selection because we are now evaluating more individuals. In this mega environmental designs, we can evaluate instead of 20 genotypes, we can evaluate 150 genotypes with the same resources, or 102. So we're playing with the selection intensity. We're also changing the accuracy with which we evaluate those individuals, and then time. Other things that we can do to improve the efficiency and the precision is improve the phenotyping, right? The least variability with that we have for a phenotype, the better, because our variabilities are going to be higher. We're going to be able to have a better estimate of those genotypic mean values. And so working with improving the phenotyping is a good idea. We just show you how to do that with experimental design. You can also do that with aided technology. And so we can improve that with image analysis and try to figure out other traits that maybe we were not able to do it individually. For example, panicle architecture traits. One of my students is evaluating the panicle architecture node, trying to figure out which of these panicle architectures provide the highest highest yield, which ones of this can we phenotype properly, and then also grain characteristics. Some of those to be used as indirect selection, but some of those to be used as direct selection because of the industry requirement of the grain size and shape. And so different strategies that can help us improve the phenotyping are also very good to include. And so for this section, I told you about different strategies that we could use to improve the speed, the efficiency, and the precision. And we're going to have, this is just a product placement. We're going to have in the agronomy meeting, so ASA, CSA, and Soil Science Society meetings. We're going to have this year, the conference is going to host a symposium, oral and poster sessions on the science for predictive agriculture. So some of the things that I told you we were looking at, we're going to have invited speakers, and then we're going to have volunteer. So if you are working in these areas, please submit to those. And if you have ideas of who do you want to see as speakers, let me know, because we're in the planning phases of the symposiums and oral poster sessions. Good. So the other thing that I told you at the beginning is we need to think about this multiple breeding targets, and we have all these new things that people care about, not just grain yield. And so the society is more interested in this cropping system performance, and we are having a higher demand for healthy and nutritious food, but also we want profitability, of course, for the farmer. And I'm putting profitability and not just grain yield, because we can get there in different ways, right? Maximum yield doesn't guarantee profitability, but I think that thinking about profitability is something that we need to start doing more seriously. We need to support those farmers' communities. There is a lot of research showing that by supporting the communities, we have a healthier environment for everyone, and then that's gonna be a benefit for the whole society. We also need stability and resilience, right? We're having this higher climate variability. It doesn't mean that we're gonna get warmer and then we can just select for warmer climate. It means that we're gonna have more extreme environments, and that we will need to be able to select for genotypes that will be able to withstand those that variability and still produce good performance. But also, we're interested in soil health and nutrient insufficiency, weed suppression, crop protection, soil microbiomes, water quality. So there are all these other dimensions that we need the agricultural systems to provide. So we need the systems to be able to maintain the soil, to be using the nutrients very efficiently, to suppress the weeds so that we can reduce the chemical use, have crop protection so that we can again reduce the chemical use, to protect that soil from many standpoints, but also the biological and the microbiomes, and also water quality. And you have seen in Iowa probably how the society is going to ask the farmers more and more that that the water quality that they drink is not being poisoned or affected by the production system. So we as a researchers need to come up with solutions that will be able to provide that so that we can keep producing food that better. Also, we need crop rotations and winter cover to help in this systems. So how do we tackle this? How do we tackle all of this multiple breeding targets? And breeders have been breeding for multiple traits since the beginning, right? I don't know any breeder that only look at one trait. We always need disease resistance. We always need maturity. We always need a lot of other things besides just grain yield. So a few strategies is we can use multiple traits in a very smart way, right? Here I'm showing you one study where we were looking at using multiple traits to improve the predictive ability of a trait that was, this is bread quality, one of the bread baking qualities, that is very expensive and it takes a lot of time to measure. So our idea was, and then breeders cannot afford to evaluate all the individuals early in their population for that trait. So, so one option is we can predict just those single traits with genomic prediction, but what about using other traits that maybe are simple, easy to measure, inexpensive? How can we improve or would we improve the predictive ability? So our strategy was including, this is just single trait prediction, and this is for different traits. This is a classic CV1 type of cross-validation. Then we evaluated, how about, I'm gonna go here first, how about we use two traits? So the second trait being fully evaluated in all the individuals. So now we're relying on that correlation among the traits to improve the predictive ability. So in that case, you can see that the predictive ability increases a lot. It does require more phenotyping, right? The other thing is, how about for traits that are post-harvest, right? Like in this baking quality, what we have is we have harvest the whole experiment, but we can decide which ones to be sent to evaluate for the very expensive traits and which ones for the other traits. So what if we purposefully create this imbalance in such a way that we will be evaluating maybe different individuals or different genotypes for the different traits so that we can enrich our correlation structure and predictive ability. So in that case, you can see that by having the same level of phenotypic evaluation as in this one, as in the CV1 of the one trait, we were able to increase the predictive ability by a lot. So it was a significant increase there just by deciding who do we send to phenotype for what trait. So we're creating a little bit of imbalance. We're not phenotyping just a small set of individuals for all the traits, but we're deciding who goes being evaluated for each one of those. This works well for those type of traits where you're still gonna have the yield trials there, but you're gonna do the post-harvest in just some of them. By thinking about those wisely, we can increase the predictive ability. Other things is we're thinking about this replace phenotyping in another case. So this is for barley, where molting quality traits are very expensive and also difficult to measure, or they take time to measure, and then the capacity of the labs is quite small compared to what a breeder would need. And so the same situation as in the wheat, what we evaluated again is using this other correlated traits to improve their predictive ability. And you can see how we can increase that by a lot by using this correlated traits. So again, thinking about genomic prediction in many different ways. We can use it for different strategies. And one of those could be to think about what to phenotype a little bit more smart, and use this replace phenotyping instead. Other things that are more compound, right, that include multiple dimensions of the traits is resilience and stability. So this is one study that is not published yet, in which what we're doing is we're looking at this historical information for oats, where we have more than 20 years of data, and we have the performance in more than 6 locations of different oat genotypes. And we looked at how stability was for those different genotypes, and then resilience, and tried to map those. And what we found is that there were different regions responsible for stability and resilience, which is not unsurprising if you think about the physiology behind those. One thing is to be able to perform well in the common sets of environments that we get, or this subset of all the possible environments. And the other one is, how do we do on a crisis compared to a normal year? So, so we were able to map different regions, and we're trying to figure out a little bit more about those regions before we publish this. But this is another thing that we need to think about. And then how do we design experiments so that we can evaluate those fast without having to wait for 20 years of data before we release a genotype, right? So those things are very important to consider. So we're trying to map those so that we can use those as proxies for selection. Other dimensions that are important is all the stakeholders that are involved, right? If we're commodity breeding, then that becomes more restricted, but we're now seeing more and more interest in the whole society getting involved. So this is just one example where we're breeding for artisan bread baking quality here in Wisconsin, and we have involved the bakers. So we have a set of bakers who are testing our advanced materials before we release them. We're also working with the farmers. The farmers come and visit our trials, and we decide with them, and then we do on-farm trials so that we can decide on whether those are gonna work well. We do baking tasting with the general public and to see whether they are gonna like those products, so that by the time we release a variety, we have the whole chain of stakeholders involved and ready to adopt the technology. And that's another thing that is important when you're thinking about breeding strategies, is to involve all those who are going to be affected by this. And then finally, I told you about cropping systems, right? And so the question becomes, can we breed for cropping systems? And I think that this is what's gonna define the next years in breeding, is can we actually stop thinking about my crop in isolation? The crops are not grown in isolation. They are grown in a landscape in the same time, spatially related, but also they are grown related in time, right? So we do plant, I don't know, one common rotation that is very good in Wisconsin is a small grain, a corn, a soybean, and then back to the small grain, or a forage there. So those type of rotations have been shown to be more beneficial for the farmers in terms of profitability, in terms of the, and then in terms of the environmental benefits of those. So there are many reasons why they do the crop rotations. So then the question remains, why do we keep breeding for them independently? Why don't we think about the whole system? So the questions are, can we have a node that is going to be better as a rotational crop in my maize? Or can we have a maize that is going to be better performer in the rotation, and the same for soybean? So there is evidence that there are genotypic differences, and the different genotypes affect some of those. We have some evidence for that they affect the next crop in the rotation. So can we start breeding for those and thinking more broadly about our breeding targets? Again, for that, we need to add all these other dimensions, right? We need to add all the dimensions on this crop rotation to make it work for the farmer and the whole society. So another product placement, we're going to have a symposium on the agronomy meetings on breeding for systems, resilient crops and ecosystem services. Again, if you have speakers you would like to see, if you're working on this, I encourage you to submit your work for oral or poster sessions. There's also going to be a poster competition for students too. So just please register for those. The abstracts are open now at this time, so you can submit that. And finally, I want to finish with this slide of we need to think about this transdisciplinary teams. Breeding is not just a one-person job. It's a multiple dimensions, multiple people job involvement, so that we can reach all of those multiple targets that we need to get. So think about that when you are doing your training as a student. Think about what are the skills that you're going to need, but people skills is going to be one that is going to be very important, but also being able to integrate all of this different dimensions and all of the different disciplines is going to be very relevant for your job. So, so this is my closing statement. I do want to leave a slide with the funding and acknowledgements. This we work with a lot of people, and we do have a lot of collaboration. With that, I'd be happy to take questions. Thank you.